You have probably noticed that children, or offspring, resemble their parents in many ways. This is the result of DNA, the hereditary substance of life, being passed from one generation to the next. Within DNA lie units of heredity called genes. Genes help determine the physical traits of a person. In this program, you will learn how genes are expressed. That is, how the information encoded in DNA is used to build proteins. An organism's physical traits make up its phenotype. In the early 1900s, the concept that genes affect an organism's phenotype was widely accepted among scientists. However, the question of how exactly genes affect traits had not been determined. The first person to investigate how a gene determines an organism's traits was the English physician, Sir Archibald Garrod. Garrod suggested that genes determined phenotypes through enzymes. Enzymes, as you might recall, speed up chemical reactions within a cell. Garrod supported his hypothesis with his studies of the hereditary disease alkaptonuria. Victims of this disease excrete urine containing the chemical alkapton, which causes the urine to appear black. Garrod hypothesized that alkapton builds up when the enzyme needed to break it down is not present. He claimed that the absence of this enzyme is the result of a defective gene. Garrod published his ideas in 1909, but they were ignored for several decades. In 1941, American scientists George Beadle and Edward Tatum produced the best evidence that genes control the production of enzymes. Through experiments with the red bread mold, Neurospora crassa, Beadle and Tatum were able to conclude that one gene controls the production of one enzyme. This is called the one gene, one enzyme hypothesis. Enzymes, as you may already know, are proteins. All proteins are made of polypeptides. A polypeptide is a long chain of amino acids. It has been discovered that each polypeptide in a protein is controlled by its own gene. Thus, Beadle and Tatum's one gene, one enzyme hypothesis has been changed to the one gene, one polypeptide hypothesis. Before we go on to explain the mechanism by which a gene controls the assembly of a polypeptide, let's review the structure of deoxyribonucleic acid, or DNA. The DNA double helix resembles a twisted ladder, which spirals from end to end. Each side of the ladder consists of a chain of sugar phosphate groups. The rungs of the ladder consist of pairs of nitrogen-containing, or nitrogenous, bases. There are 4 types of nitrogenous bases found in DNA. These are divided into 2 groups, purines and pyrimidines. The bases adenine and guanine are purines, while the bases cytosine and thymine are pyrimidines. Adenine only pairs with thymine, and cytosine only pairs with guanine. A single base, together with a phosphate group, and deoxyribose, the sugar component of DNA, make up a nucleotide. A nucleotide is the basic unit of DNA. Genes are hundreds or thousands of nucleotides long. Recall that a gene directs the production of a polypeptide. Since a polypeptide is made up of a chain of amino acids, a gene must control the linking of the amino acids. Scientists have determined that the order of the bases of 3 adjacent nucleotides of a gene is the code that directs a particular amino acid to be added to a polypeptide chain. Each 3-nucleotide sequence is called a codon. Remember that in a eukaryotic cell, DNA is confined to the nucleus. Polypeptides and proteins, however, are assembled in the cytoplasm of the cell. Knowing that genes direct the assembly of polypeptides, you are probably wondering how a gene is able to direct the assembly of a polypeptide when the gene is in the nucleus and the polypeptide is assembled in the cytoplasm outside of the nucleus. The solution is ribonucleic acid, or RNA. There are only two differences between the chemical makeup of RNA and the chemical makeup of DNA. In RNA, thymine is replaced by the nitrogen-containing base uracil, and the 5-carbon sugar is ribose rather than deoxyribose. Structurally, RNA differs from DNA in that it consists of a single strand of nucleotides, whereas DNA is double-stranded. To form a protein from DNA, two processes are involved: transcription and translation. Transcription takes place first and is the process by which the DNA code for a polypeptide is copied by RNA. Translation is the process by which the message transcribed by RNA is converted to a polypeptide. Before RNA can copy the DNA code, it must gain access to the code. This is done with the aid of an enzyme called RNA polymerase. RNA polymerase binds to DNA and unzips it. That is, it separates its two strands. A single, separated strand of DNA with its unpaired nucleotides now serves as a template for RNA. Next, RNA nucleotides assemble along the exposed DNA nucleotides by matching up with complementary bases. This nucleotide assembly continues until a certain stop code is reached on the DNA molecule. Finally, the newly formed RNA strand, which is a transcript of the protein building instructions encoded in DNA, breaks away from the DNA strand. An RNA molecule that copies the DNA code for a polypeptide is called messenger RNA, or mRNA, because it serves as a genetic messenger between DNA and the protein-making machinery within the cell. Each group of 3 nucleotides on the mRNA molecule makes up a codon. Each codon specifies an amino acid. Thus, the sequence of codons along the mRNA strand will be decoded into a sequence of amino acids, forming a polypeptide chain. In eukaryotic organisms, such as ourselves, messenger RNA undergoes a few alterations in the nucleus before traveling to the cytoplasm. In eukaryotes, the protein coding segments of genes do not lie next to each other on a chromosome. Instead, they are separated by non-protein coding gene segments called introns. The segments of each gene that code for a protein are called exons. Both introns and exons are transcribed by messenger RNA. Next, a cap is placed at one end of the messenger RNA transcript and a tail, called a poly-A tail, because it consists of about 200 adenine nucleotides, is placed at the other end. The cap and tail prevent the messenger RNA from being broken down by enzymes. Once the cap and tail are placed on the mRNA transcript, the introns, or non-coding segments, are cut out and the exons are spliced together. The mRNA is now ready to move into the cytoplasm. or it will be translated into a polypeptide. Messenger RNA is only one of 3 types of RNA. Another type is transfer RNA, or tRNA. Like mRNA, tRNA is transcribed from a DNA template in the nucleus of a cell. The tRNA molecule is typically much smaller than mRNA. While mRNA may be 1,000 or more nucleotides long, tRNA has only about 80 nucleotides, which assume this three-dimensional shape. Notice that at one end of the tRNA molecule there is a short tail. This tail serves as the amino acid attachment site. At the other end of the tRNA molecule, there is a loop of nucleotides. Within this loop lies a sequence of 3 bases which make up an anticodon. An anticodon is the complement of an mRNA codon. The anticodon in this case consists of one molecule each of uracil, adenine, and cytosine. Since uracil always forms a pair with adenine and cytosine pairs with guanine, the codon on the mRNA molecule must consist of one molecule each of adenine, uracil, and guanine. This codon specifies for the amino acid carried by the tRNA molecule. Thus, tRNA acts as a vehicle by bringing a certain amino acid to a certain place specified by mRNA. The third type of RNA is called ribosomal RNA, or rRNA. Like mRNA and tRNA, rRNA is also transcribed from DNA. Within the nucleus of a eukaryotic cell, there is a structure called the nucleolus. Inside the nucleolus, rRNA is joined with various proteins to form ribosomes. Each ribosome consists of 2 subunits that are unequal in size. The ribosome is the site of protein synthesis. As stated earlier, the synthesis of the 3 types of RNA and the assembly of the ribosomes occur within the nucleus of the cell. The RNA and ribosomes then pass from the nucleus to the cytoplasm via pores in the nuclear envelope. Within the cytoplasm are all the amino acids. The cytoplasm is the site of translation, the process by which the information transcribed by mRNA is converted to a polypeptide. In the cytoplasm, amino acid molecules become attached to their specific tRNA molecules. This creates a pool of tRNA molecules with amino acids in tow. Next, ribosomes become attached to mRNA at various sites along the strand. The small ribosomal subunit is the first component of the ribosome to become attached to the mRNA strand. At the same time, a tRNA with the anticodon UAC attaches to mRNA at the site of the complementary codon AUG. AUG is an initiator codon, that is, a codon which starts translation. Once the tRNA has attached itself to the mRNA at the site of the initiator codon, the large ribosomal subunit joins the complex. Notice that there are 2 groove-like sites in the large ribosomal subunit, the P site and the A site. Into these fit the tRNA molecules carrying amino acids. The large ribosomal subunit joins the complex so that the initiator tRNA fits into the P site. This leaves the A site free for the next amino acid carrying tRNA molecule. Once the initiation of translation has taken place, an amino acid carrying tRNA molecule with an anticodon complementary to the next codon on the mRNA strand moves into the vacant A site. A peptide bond links the amino acid of the A site to that of the P site, forming a dipeptide. Once the dipeptide is formed, it separates from the tRNA in the P site, but remains bound to the tRNA in the A site. Having fulfilled its duty, the tRNA leaves the P site ready to pick up another amino acid. The mRNA moves along the ribosome until the tRNA carrying the dipeptide occupies the P site. The A site is once again free to accept another tRNA molecule carrying an amino acid. Molecules of an amino acid carrying tRNA continue to enter and leave the ribosomal sites as the mRNA moves along the ribosome. Thus, the dipeptide becomes a polypeptide. The polypeptide grows in length until a stop codon is reached, at which time the A site of the ribosome accepts a protein called a release factor instead of an amino acid carrying tRNA. The release factor causes the last tRNA in the P site to exit the ribosome. At the same time, it breaks the bond between the polypeptide and tRNA. The polypeptide goes on to coil and fold itself into a functional protein. Finally, the two ribosomal subunits detach from the mRNA. As an organism develops, some genes are expressed, or turned on, in cells, while others are turned off. Thus, different proteins are produced during different stages of development. But how does a cell know when to turn on a gene? While biologists have come to understand this process in prokaryotic organisms like bacteria, what turns genes on and off in eukaryotic organisms, such as this caterpillar, is still largely a mystery. This concludes our presentation. By now, you should have a general understanding of how a gene is expressed. That is, how a gene controls the production of a protein.